Okay, so we're going to continue with our lesson that we started with looking at mutations. So this is more like a continuation of what we looked at yesterday when we talked about mutations. It's just a continuation of that. We did mention that mutations can either be spontaneous or they can be non-spontaneous. And if the mutations are non-spontaneous, these mutations come about due to substances
Chemical.
and due to what are known as mutagens. So non-spontaneous mutations come about due to what are known as mutagens. And so today's lesson is concentrating on mutagens and their actions. Mutagens and their actions. So I'm going to have an introduction, and in the introduction, we're going to see that we talked about what a mutation is as a permanent change in the DNA, in the gene, in the case of the gene mutation, or it could be a chromosomal mutation, which occurs in the entire chromosome. So a mutagen is any substance or agent that can cause a mutation or a change in the structure or sequence of DNA. So mutagens cause non-spontaneous mutations. These mutagens are going to change the genetic makeup of an organism by changing the DNA. And usually these mutagens are carcinogenic in nature. Most mutagens are also carcinogenic. In short, they predispose cells to have cancer. They predispose cells to have cancer. So mutagens are usually carcinogenic. And then common mutagens that we have is ethidium bromide, formaldehyde, dioxin, and nicotine. So these are some examples of mutagens that that are there or that exist. These are some of them. So we did talk about what mutations are, and we said that a mutation could be a replacement of a nitrogenous base with another in one or both strands. So it could be a base substitution mutation, or it could be a frame shift mutation, that is an addition or deletion of a base pair in a DNA molecule. So a mutation will lead to the change in the genetic makeup of an organism. We did look at mutations last time. Mutagenicity simply refers to the induction of permanent changes in the information content of the genetic material. So mutagenicity simply from a similar word of mutagen. So this is just a process where there is an induction of a permanent change in the information content of the genetic material. So we talked about mutations last time. We said that mutations could be either changes in the genes or changes in the chromosomes, either changes in the genes or in the changes in chromosomes. And gene mutations, which are of particular interest for our course, they could be due to substitution or due to frame shift or addition or deletion of a nucleotide base. And then we have chromosomal mutations. These confer changes could be in the number of chromosomes or in the structure of the chromosome itself. So mutations can also be categorized whether they are gene mutations or chromosomal mutations. So this is just depicting a parental DNA that has been altered, and we're going to look at this process, but take note of this molecule. And this is going to confer an altered parental DNA, and through replication, we're going to find that there could be an altered daughter DNA, and that will confer a mutated granddaughter DNA, an altered granddaughter DNA. So it's a process since information is passed from one generation to another. Now, what are the effects of mutagens? What do they confer? So mutagens are genotoxic in that they're harmful to DNA in a variety of ways. So they could be either direct or indirect ways that they affect. So if it is at the chromosomal level, as I mentioned, the mutagens can alter the number of chromosomes or the structure of the chromosomes. So there are a number of chromosomal abnormalities that can be produced by these mutagens. So remember, they could either be number or they could be either be due to the structure. So there could be a deletion, there could be an addition of bases, or there could be a deletion of a segment of a chromosome. There could be an insertion of a segment of a chromosome. There could also be duplication of a segment of the chromosome. There could also be translocation, monosomy, which has to do with the number of chromosomes. We have also non-disjunction as some of the abnormalities that are produced by the mutagens in the chromosome. And then these mutagens can also affect or dysregulate the central dogma of molecular biology process as depicted on the diagram there. It can affect DNA replication, therefore an error can occur there. It can affect the transcription process. It can affect the translational process as well. So they can also affect the central dogma of molecular biology. And at the molecular level, these mutagens can create different gene mutations that can end up in the loss of function of a protein in an altered function or a non-functional protein. So their errors can occur within the protein when it folds into its final shape. So these are the mutagenic effects, the effect that mutagens confer. Now, what are the sources of mutagens? What are the sources of mutagens? So mutagens can be chemical, they could be physical, they could be biological. So we have physical mutagens, which of examples are heat and radiation. Heat and radiation. And examples of radiation, we have UV or ultraviolet radiation, which could be both from natural sunlight and also from turning birds. And then we have X-rays, which are employed in medical, dental, airport security screening. Then I mentioned that the other sources of mutagens could be chemicals. So for example, in cigarettes, these contain dozens of mutagenic chemicals, and hence you find that someone can have or can develop cancer because of cigarette smoking. We have other chemicals such as benzoyl peroxide, which is found in main acne product. We have other mutagenic substances or chemicals. And then we have biological mutagens. And of importance, we have infectious agents. So for example, viruses, other examples include bacteria as well. So for example, Helicobacter pylori, which is a bacteria that is spread through contaminated food. We also have an example of human papillomavirus, which is transmitted through sexual intercourse. So these are the sources of mutagens. So I've already explained this process of mutation that can occur during the process of replication. So mutagens can have an effect on the replication, transcription, and also translation process. So now let's look at these types. As mentioned, I've mentioned that there could be chemicals. Now, under chemical mutations, there could be alkylating agents. We have alkylating agents. We have base analogs. We have methylating agents. We have DNA intercalating agents as part of the chemical agent. And then we have also reactive oxygen species. We have also physical mutagens. We mentioned UV light and heat as well. And also part of radiation could be UV radiation or ionizing radiation. So UV radiation is non-ionizing. So it could be ionizing or non-ionizing radiation. So let's look at the physical mutagens. So the physical mutagens, we mentioned that they could be non-ionizing radiation and ionizing radiation and also heat. So radiation and heat are examples of the physical agents, physical mutagens. So now looking at the non-ionizing radiation, we have UV light. So UV light causes what is known as dimerization or dimerization. So it leads to the formation of a pyrimidine dimer, and that dimer causes a kink or a bend in the DNA molecule. So energy, the UV light is a physical mutagen whose energy causes adjacent thymine or cytosine bases in the DNA to covalently link together, forming a dimer. And this process is what is known as dimerization. So what will happen is that there's a formation of thymine dimers. In the case of thymine, as depicted there, there will be formation of the dimers. Now, these dimers are going to cause the strand to buckle. And when that strand buckles, this is going to disrupt the normal base pairing. Remember, there's a normal base pairing. A adenine should base with thymine. However, because of this disruption, this formation of dimers, this disrupts the normal base pairing. And then this hence means that there's a prevention of normal process of replication and transcription. So these processes are actually disturbed due to this non-ionizing radiation of UV light. So this is simply illustrating the formation of a thymine dimer, which leads to the premature termination of the complementary strand. Remember, this is a normal DNA, and through UV light, you find that thymine and thymine here, due to the effect of the UV light, they form a dimer. They are linked. They are joined together here. They become joined, making a T-dimer. Now, when this is made, it means that this complementary strand cannot continue to be synthesized. It cannot go on. So there's a premature termination of the complementary strand. So this is the effect of UV light, which is non-ionizing radiation. So this is to explaining the same, the formation of dimers. So it's possible the enzymes can be able to cut out and remove this damaged DNA. And when that is removed, the polymerase is then able to fill in the gap by synthesizing new DNA using an intact strand as a template. Then just as in the process of replication, the DNA ligase will then seal the remaining gap by joining the old and the new DNA strands. So that's UV light. The other one is ionizing radiation. Ionizing radiation will compose of gamma rays and X-rays as well. Examples include X-rays and gamma rays. And sources include artificial sources and also natural sources. So under the natural sources, there's X-rays, these gamma rays, these ionizing radiation can come from sunlight, can also come from outer space. And then we have the artificial sources, which could be medical diagnostic. Remember, I gave an example of X-rays being used in medical diagnosis. For example, chest X-rays. If someone is sick, you're going to cut out an X-ray. So that could be sources of ionizing radiation, and also power plants as well. So how do ionizing radiations cause mutations? How are they mutagens? So these produce what are known as reactive free radicals. They produce highly reactive free radicals. And these produced radicals, what they will do is that they are going to react with biological molecules. So there will be an interaction between these reactive free radicals with the DNA, the proteins, and the lipids in the cell membrane. Right? So they react with the DNA, they react with the proteins, they react with the lipids in the cell membrane. And these radiation hence will lead to damage in the base and the sugar residues of DNA, for example. Remember, DNA is composed of nitrogenous bases, sugars, and also phosphate or phosphoric acid. So it's going to damage the base and the sugar residues in the DNA. So we have cells that are most affected by ionizing radiation. Actually, they're rapidly dividing cells. So for example, the bone marrow, cells of the bone marrow, cells of the GIT, these are mostly affected by ionizing radiation. And how severe that effect will depend upon how much of the radiation that cell has received. So these are the ionizing radiation. So this is how they interact. And in the interaction with DNA, this is what they are going to do. Through the production of the free radicals, and also as a direct effect, they lead to a break in one or both of the DNA strand. They're going to lead in the breaks in one or both of the DNA strand. Now, this can lead to a rearrangement, can lead to deletion, lead to the loss of the chromosome. And hence, if this action is unrepaired, what will happen is that death will occur. There will be death occurring. These ionizing radiation also cause damage to and loss of bases, as explained earlier. They're going to damage the bases, leading to a loss in the bases and hence causing mutations. They also cause cross-linking of DNA to itself or to proteins. So due to their effect, what will happen is that DNA is able to cross-link to itself, and also they can cross-link DNA to proteins as well. So what are the effects of this whole process having happened? So as mentioned earlier, these mutagens will lead to a loss of cellular function, So there'll be a disruption in the function of the cell. The other things that an organelle would fail. There could be organelle failure. Remember, the organelles are in the cell. So organelles, a certain organelle can fail due to these ionizing radiation. The other thing that they can lead to a blockage in the cell division. Cell division can be blocked. And then, as mentioned earlier there, it can lead to the death of the cell. The death can result. So these are the effects, ultimate effects of the ionizing radiation interaction with DNA. So this is simply summarizing what radiation does, whether ionizing or non-ionizing radiation. Formation of dimers, in the case of UV light, we mentioned that. We did mention that ionizing radiation can cause cross linkage of DNA as in this case. So there could be cross linkage of the DNA, as in this case. We also mentioned that ionizing radiation can cause base change. The bases can change, or it can cause the loss in the base. Can also, we mentioned that these can also cause a physical breaking in the strands. There could be hydrogen bond breakage there among the bases. Can also cause double strand breaking of the DNA, or even a single strand breaking. We also mentioned that there could be a link between DNA and the protein. So we have a protein cross linkage as well. So these are the physical mutagens of radiation. The other mutagen, physical mutagen, is heat. So when heat is heated to a specific temperature as above 95 degrees Celsius, we find that the DNA double strand is actually denatured. So by denaturization, the two strands of DNA are separated. They are separated. And then also heat stimulates water-induced cleavage of the N-glycosidic bond. So this is a bond which attaches the base to the sugar compound in the nucleotide. So heat will cause denaturization. It will also induce cleavage of the glycosidic bond between the sugar and the base. So this mutation heat mostly affects the purines, and when that is done, the process is known as depurination. Depurination, and so there's removal of purines, depurination, and also in the pyrimidines it can happen, and this will lead to a process known as depyrimidination. So depyridination and depyrimidation. So this can result in a baseless site. Remember, due to the process of depurination and depyrimidation, what will happen is that there's a loss of the purines or the loss of the pyrimidine, which means that point where there was a purine, what will result is a baseless site. So there will be a baseless site at that point where there was a purine or where there was a pyrimidine. So there will be that gap. So this reaction is usually not mutagenic because cells are actually able to repair that mechanism for gaps and the nicks. So it's repairable. It's repairable. So this is just illustrating depyrination as explained earlier. So that's about heat being a mutagen. And then we have the chemical agents. We have the chemical agents. So among the chemical agents, we have different chemical agents. There could be terminating agents, alkylating agents, base analogs, as mentioned earlier, bulk addition products, or intercalating agents. These are some of the chemical mutagens that we have. So chemical mutagens, let's look at the chemical modification agents. We have what are known as chemical modification agents. So these compose chemicals which alter the structure and the pairing properties of the normal bases. They alter the chemical, the structure and the pairing properties of the normal bases. So of these are active on both the replicating and also the non-replicating DNA. These are active on both replicating and non-replicating DNA. Hence, these chemical modification agents will result in a mutation on the DNA replication, leading to the formation of baseless sites or a mispair. So they're going to lead to a formation of baseless sites or even mispairing of the bases. So there are two common chemical modification agents, and these are alkylating agents and deaminating agents. So two common chemical modification agents are alkylating agents and deaminating agents. Now, the chemical modification agents of deaminating agents in this case, these cause oxidative deamination of amino group in adenine, guanine, and cytosine. They're going to cause deamination of the amino group that are found in adenine, guanine, and cytosine. An important example is nitrous acid. Remember when we started, I showed the picture and I said that pay attention to this molecule. That's nitrous acid. It's a very common deaminating agent. So what nitrous acid does is that it converts the amino group, as mentioned above, into a keto group. So it converts an amino group into a keto group. And this also leads to a change in the hydrogen potential of the bases that have been modified. So nitrous acid will convert the amino group into a ketone group, and hence this changes how the hydrogen bonding is going to occur in these modified bases. Okay, so these are some of the actions of the mechanism of nitrous oxide.
Hello, sir.
Hello?
Yes, sir. Would you please go to the previous page so I can take a screenshot?
I'll share the notes.
No, right, thank you.
Yes. So these are the mechanism of, this is the mechanism of nitrous oxide. So we have guanine, remember I mentioned guanine, cytosine, and adenine. So it's going to change the amino group into a ketone group. So in this case, we have guanine here. Guanine, through the mutagen of nitrous acid, converts this guanine into xanthine. So this is xanthine. Remember, this is the NH2, which is the amino group. It has been converted to a keto group, which is xanthine. And then we have cytosine here. And remember, we have the amino group there. Through this mutagen, the mutagen is going to cause this amino group to be converted into the keto group here, And then adenine, in the presence of this mutagen of nitrous acid, is going to convert this amino group into the keto group. And what is formed is hypoxanthine. You have hypoxanthine. So adenine is converted to hypoxanthine. Guanine is converted to xanthine. Cytosine has been converted to uracil. So this is what nitrous acid does. This is what nitrous acid does. This is the mechanism of nitrous acid. Or these deaminating agents, they convert an amino group into a ketone group, and this affects the way the hydrogen bonding is supposed to happen. Remember, in the case of guanine has to bond with cytosine with three bonds. Three bonds are supposed to form between guanine and cytosine, but because of this mutation that has occurred, because of the presence of this mutagen of nitrous acid, the base pairing has been disrupted. The hydrogen base pairing has been disrupted. Okay, then we have the chemical modification agents of alkylating agents. So these modify the normal bases by adding alkyl groups. Are going to add alkyl groups. No wonder they are called alkylating agents. And common examples are ethylmethane sulfonate, or abbreviated as EMS, or nitrosoguanidine, abbreviated as NG, or di-2-chloroethyl sulfide, or sulfur mustard. The other example is di-2-chloroethyl methylamine, which is simply nitrogen mustard. So these are some examples of the common alkylating agents, and this is EMS or ethylmethane sulfonate. This is the structure of EMS. So these modify by adding alkyl groups. So the mechanisms, and in this example, we are giving EMS, which is ethylmethane sulfonate. We have guanine. In the presence of this mutagen EMS, is going to lead to the formation of 6-ethyl-guanine. 6-ethyl-guanine. There's been an addition of this alkyl group. There's been addition of this alkyl group, forming 6-ethyl-guanine. 6-ethyl-guanine. And this is another example. In the thymine, through the presence of this mutagen of ethylmethane sulfonate or EMS, leads to an addition of the IQ group. That led to the addition of an IQ group. So these diagrams are simply illustrating the mechanism of what we've already talked about, the addition of IQ groups. And then chemical mutagens, we have other types known as base analogs. So an analog resembles a certain structure in a reference. So in terms of these base analogs, these chemicals structurally resemble the purines and the pyrimidines. And so since they have that resemblance, they may be incorporated into the DNA in place of those normal bases during the process of DNA replication. So base analogs, very simple. You have a purine which is supposed to be in a DNA molecule, but we have this base analog because it is similar to a certain purine, which is going to be incorporated into that place where that normal base was supposed to be. So it's known as a base analog. So what will happen is that they are going to lead to incorrect insertion of nucleotides opposite them in replication. They are going to lead to an incorrect insertion of nucleotides opposite them in replication. So examples of base analogs, two important examples are 5-bromouracil and 2-aminopurine. 5-bromouracil and 2-aminopurine. These are important examples of base analogs. So let's look at this common one, this common agent of 5-bromouracil, which is an analog of a pyrimidine. It's an analog of a pyrimidine. It is actually extensively explored in research. It is an artificial and it resembles thymine. However, it has a bromide atom instead of a methyl group. It has a bromide instead of a methyl group at position at carbon 5. So this bromouracil will be incorporated into the DNA and hence it's going to pair with adenine, just like thymine does. So it is incorporated in DNA and then it's going to pair also with either adenine, as does thymine based pairs with the adenine, or due to the process of tautomerization or isomerization, can pair with also guanine. So the mechanism, so this is the same tautomerization, which is spontaneous structural alteration between two forms. That's tautomerization. So there's an alteration between two forms. It can occur in a keto form or in an eno form, as you are going to learn in your organic chemistry. So